We're doing the exercise Gene Regulation, Human INSIG1 and BLAZE. The first part of the question asks, find the location tab, a region in detail page, for the region between the genes INSIG1 and BLAZE. Are there any predicted enhancers in this region? As we are interested in human genes, I'm going to select human from the species selector drop-down list. We're asked to find a genomic region between two genes. To do this, we could search for either INSIG1 or BLAZE. I'm going to search for INSIG1 by typing INSIG1 into the search box and clicking Go. You can see our gene of interest at the top of the search results. To go directly to the Location tab, Region in Details view, please click on the gene genomic coordinates shown in green under the gene name. The second image on this page shows 1 megabase region around our gene of interest. As you can see, it's centered around INSIG1, shown as a yellow bar in the genes track, also indicated by the red box. You can find a BLAZE to the right, also shown as yellow bar. As we are interested in a genomic region between these two genes, I'm going to drag out a box spanning both INSIG1 and the neighboring BLAZE gene. This opens a pop-up menu with an option to mark this region or jump to it. I'm going to jump to the new selection, which is going to refresh the image below. Let's now scroll down to the regulatory build track, showing regulatory elements in this region. Colors are explained in the legend below, with enhancers shown as yellow boxes. There is only one yellow enhancer in the region between these two genes. The next part asks, go to the Regulation tab for the enhancer ENSR 00011333586. How many cell types is this enhancer active in? Are there any cell types where its activity is repressed? Let's now click on the enhancer to get a pop-up menu with its ID and other information. Click on this table ID ENSR 00011333586 to open the Regulation tab Summary view. Scroll down to see the summary of regulatory activity across different cell types shown as a table on the bottom of the page. As you can see, this enhancer is active in only one cell type, neutромyelocytes, and it's repressed in 34 different cell types. The next question asks, go to the Details by Cell Type page. Take a look at the histone modifications across this enhancer in neutромyelocyte cells, where this enhancer is active, compared to neutrophil CB cells, where it's poised. What differences can you observe? To do so, let's scroll up to the top of the page and click on Details by Cell Type icon, which can be also found in the left-hand side menu. Let's add data to this view by clicking on Configure Cell Tissue blue button. Choose cells by ticking a box next to neutромyelocyte and neutrophil CB. The cells have been now added to the view. Let's now add experiment tracks by clicking on the Experiments tab and select All and the histone. Our experiments have also been added to the right-hand side panel of selected tracks. Once you're done choosing your options, click on Configure Track Display, then View Tracks to load the page. Our enhancer is active and shown in yellow in neutромyelocytes, while in neutrophil CB cells, it's poised and shown in purple. The experiment peaks are shown as colored blocks with the ChIP-seq signal below. Both cell types have trimethylation of histone H3 at lysine 9 and 27, and monomethylation of histone H3 at lysine 4. While neutромyelocyte cells also have acetylation of histone H3 at lysine 27 and trimethylation of histone H3 at lysine 36. Different clusters of peaks indicate different epigenetic profiles for these two cell types, with neutrophil CB cells missing some elements needed for the enhancer activation at this locus. The last question says, go back to the summary page. Are there any verified transcription factor binding motifs in this enhancer? In what cells? I'm going to scroll up and click on the summary icon to go back to the summary view. Transcription factor binding motifs are shown as grey and black boxes in the motif features track, with verified motifs shown in black. There are 2 black markers falling in this enhancer. Click on them to find more information. The 2 motifs are both verified in K56-2 cells and bind a number of different transcription factors.